Animal, the last one, animal biotechnology. So similar structure, we start with how to culture the animal cell. But of course, now this is animal cell, so the culturing method will be different. So they are not totem-potent. They cannot just grow a whole new baby from a single cell. So everything are a bit different here. So you have to know the different differences in handling the animal cell culture. So we have something called the primary cell, we have something called the cell line, so have some idea on what they are. And then we talk a lot on the telomere because this is some restriction on culturing the animal cell. They will die eventually, and the existence of telomere is like why? Because they are some protective DNA at the end of the chromosome, but when the DNA grow, when they make copy of themselves, the telomere will shorten. They sacrifice themselves to protect the DNA, but they themselves will shorten, and when they reach a certain point that they're too short, then the cell will like stop growing, and they will eventually die, and the cell, it is something called aging. Okay, so have some idea on this. So this will limit how long we can keep an animal culture, because somehow they will die eventually unless you turn them into something called immortal cell line. Okay, so this happened in the animal cell, so some basic idea. But what is more important is, of course, producing this transgenic animal. Okay, so still a lot about the telomere. And then, so more important is producing a transgenic animal. Okay, so we can make use of the transgenic animal for research, for obtaining products, for testing some therapy thing. So how to do so? Again, we have to start with growing the transgenic animal. So you have to know how to do the cell isolation, and they are different from plant cell isolation, and you have to know about the difference. And then some extra thing you have to add. For example, there are more collective tissue among the animal cell compared with plant cell. So plant cell have cell wall. Each plant cell is very like separated. They are just they're like different rooms. They have a wall separation. But for animal cell, they have cell membrane only, so they are more stick together. They're more sticky. They also have many collective tissue between the animal cells. So we have to further deal with this collective tissue by digesting them, separating the animal cell one by one. So these are some extra things you have to consider when doing animal cell culture. Okay, so we talk about a lot here on isolating the cell by physical properties, by labeling them. So make sure you know some way about how to isolate the animal cell. Okay, adding label to them, passing them through a machine, and then detect them by some signaling, something like that. Okay. And then we really have a cell culture now. Again, very important aesthetically. Make sure everything is sterile, low contamination. Otherwise, your cell culture will be killed or outcompeted by the bacterial cell or by the fungi. So make sure this is sterile, and also know how to keep it sterile. And then cell culture. Yeah, so we also have different ways of growing the animal cell. So plant cell, we have callus culture, we have, what's the other type? Callus culture and suspension culture. So for animal cell, we also have two different types. We can do a suspension cell culture, or so they will lie in the middle of the culture medium, but we also do an adherent cell culture that they grow on the bottom. But it is not all your choice, okay? It depends on the cell type. So some of them will always stick to the bottom, but some of them can like throwing at the middle of the part. But we have different type of culture here. And the culture medium, same. You have to make sure you have everything needed in your culture medium. Nutrients, hormones, you have the culture condition well adjusted so that the growth of your animal cell is optimum. Okay. So after they grow, at some time you have to do subculturing, you have to do cell passaging so that their density will not be too high, that they slow down in their growth. Okay, so these are some really practical consideration when you are trying to culture your cell that you have to know about. Okay. And we mentioned something called the HeLa cell line. So there are min-motor cell line used in many labs. Okay, so this is like the part one, so still about the this thing.